Good morning students. In this video we are going to discuss about NEET and K-CET level coaching part on the chapter Cell the Unit of Life. Subtopics: Ribosomes, Cytoskeleton, Cilia and Flagella, Centrosome and Centrioles. Now let's enter into today's session. First question: Ribosomes are granular structures first absorbed by, let us see options. A. Robert Brown. B. George Palade. Option C. Camillo Golgi. Option D. Graham. The correct answer for this question is option B. George Palade. Okay, now let me explain this question in detail. Ribosomes are the granular structures first absorbed by George Palade in the era of 1953 under microscope, electron microscope, as a dense particles. Understood? So the correct answer for this question is option B, George Palade. Next, second question. Ribosomes are composed of, let's see options. Option A, RNA and proteins. Option B, DNA and proteins. Option C, both DNA and RNA. And option D, RNA and lipids. So the correct answer for this question is option A. Ribosomes, as we learned, they are membraneless cell organelles present in the cytoplasm. They are composed of ribonucleic acid, shortly called as RNA, and proteins. Understand? So correct answer is option A. Next, third question. Fill up the A and B with the correct answer. Now we need to identify correct answer for A, B from the given A, B, C, D. First, let us read the question. The eukaryotic ribosomes are dash, while the prokaryotic ribosomes are dash. Okay, now let us see options. Option A, 70S and 80S respectively, and 50S and 30S respectively. C, 50S and 40S respectively. D, 80S and 70S respectively. So the correct answer for this question is option D, 80S and 70S respectively. Okay, now let us see explanation. The eukaryotic ribosomes are 80S, while the prokaryotic ribosomes are 70S. Okay, here S stands for sedimentation coefficient, Swedberg's unit, and it indirectly measures the density and size of the ribosomes. Have you understood this explanation? This is very important for NEET and K-CET exam. Please remember. Next, fourth question. Choose the functions of the cytoskeleton. Option A, mechanical support. Option B, motility. Option C, maintenance of the cell shape. Option D, all the above. Let us find out the functions of the cytoskeleton. First of all, I would like to brief regarding cytoskeleton. An elaborate network of filamentous and proteinous structures present in the cytoplasm. They are collectively referred to as cytoskeleton. Okay, and one more regarding cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell, etc. So with all these functions, the correct answer for this question is option D, all the above. Next, fifth question. Name the protein-made connections where the tubules of the peripheral triplets are connected by radially. Option A, bridges. B, linkers. C, spokes. D, all the above. The correct answer for this question is option C, spokes. Okay, let me explain this one in detail. The central portion of the proximal region of centriole is also proteinous and called as hub, which is connected with the tubules of the peripheral triplets by radial spokes made up of protein. There also spokes also made up of with proteins. Understand? So the correct answer is option C, spokes. Next, sixth question. Swedberg's unit is useful for measuring the following organelle. Option A, nucleus. Option B, centrosome. Option C, ribosome. Option D, centriole. The correct answer for this question is option C, ribosome. Okay, let me explain this one once again. The eukaryotic ribosomes and prokaryotic ribosomes are differ. It means in eukaryotic cells, different ribosomes are present when we compare with the prokaryotic cells. How come it is? Let us see. In eukaryotic cells, the ribosomes are ATS. Example, nerve cell, muscle cell, bone cell, and sperm cell, etc. Prokaryotic cells consisting ribosomes, they are 70S type. Example, bacterium. Understand? So the correct answer for this question is option C, ribosome. Here the Swedberg unit, S stands for Swedberg unit and S stands for sedimentation coefficient. Now coming to seventh question. Choose the correct statement from the given below. Statement 1 and statement 2 are given. We need to identify the correct statement from the given two statements. Let's read the first statement. Cilia are small structures which work like oats. Statement 2. Cilia causes movement only in the cell, not in the surrounding fluid. Let's see options. Option A, only statement is correct. Option B, only statement 2 is correct. Option C, both the statements are correct. Option D, both the statements are incorrect. So the correct answer for this question is option A, only statement A is, or only statement 1 is correct. Why statement 2 is wrong? Then how come the statement 2 could be present? Let us see explanation for this question. Cilia and flagellum are hair-like outgrowths of the cell membrane. Okay. Cilia are small structures which work like oats and causing the movement of the cell or the surrounding fluid like water, etc. Understand? So second statement is absolutely wrong. It should be, the movement should be not only in the cell, it should be even in the surrounding fluid also. They pushes the fluid back to move the body upward or towards front. Understand? So the correct answer for this question is option A. Next, eighth question. Match the column 1 with column 2. Here are the two columns. In column 1, some specifications A to E. We should match with the column 2. Now let's match. Core of the cilia. Core of the cilia and flagellum is called as axoneme. So A is matching with Roman number 4. Cytoskeleton. Cytoskeleton helps in motility. So B is matching with the Roman number 5. 9 plus 2 array. 9 plus 2 array or arrangement of axonemal microtubules can be seen in cilia and flagella. So C is matching with the 2. Spindle fibers. Spindle fibers helps to divide the cell into two dotted cells during animal cell division with the help of a cell organelle called as centriole. So D is matching with the Roman number 1. Ribosomes. Ribosomes are also regarded as protein factories. So E is matching with the Roman number 3. So with this, the correct answer is Roman number, it means option number D. I repeat once again, A 4, B 5, C 2, D 1, E 3. Have you understood? This is very important concept for NEET and K-CET. Please make a note it. Next question. The core of the cilium and flagellum is called as. Now let us see options. Option A, axoneme. Option B, radial spoke. Option C, central sheath. Option D, linkers. So the correct answer for this question is option A, nothing but axoneme. Just now we had a discussion that the core of the cilia or flagellum is called as axoneme. It possesses a number of microtubules running parallel to the long axis. Understood? Now let us see 10th question. Pick out the correct statement from the given below. Statement 1. The axoneme usually has 9 doublets of radially arranged peripheral microtubules. Next, statement 2. And 1 pair of centrally located microtubules are present. Now we need to identify correct statement from these two statements. Now let us see options. Only statement 1 is correct and statement 2 is incorrect. B. Only statement 2 is correct and statement 1 is incorrect. C. Both the statements 1 and 2 are correct. D. Both the statements 1 and 2 are incorrect. The correct answer for this question is option C. Both the statements 1 and 2 are correct. It means, as we discussed just now, I would like to explain this one. The axonema usually having 9 pairs of doublets of radially arranged peripheral microtubules and 1 pair of centrally located microtubules. So such an arrangement of axonemal microtubules is referred to as the 9+2 array. Can be seen in cilia and flagellum. Have you understood this explanation? So the correct answer is nothing but option C. Now moving to 11th question. Find out the location for centrioles in animal cells. Let us see options. Option A, ribosomes. Option B, mesosomes. Option C, microbodies. Option D, centrosome. The correct answer for this question is option D. Now let's see explanation for this question. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. Okay, they are surrounded by amorphophallus pericentriolar material. Both the centrioles in a centrosome present perpendicular to each other, in which each has an organization like an cartwheel. So 90 degrees arrangement can be seen to the centrioles in one single centrosome. Understand? So the correct answer for this question is option D, centrosome. Very important area for competitive examinations. Please remember it. Next, 12th question. How many centrioles are present in the centrosome of animal cells? This area is also very important for competitive examinations. Let us see options. Option A, 4. Option B, 3. Option C, 2. Option D, 1. The correct explanation for this question is option C, 2. Explanation, as I told you just now, centrosome is an organelle usually containing 2 cylindrical structures called centrioles, and they are surrounded by amorphous pericentriolar material, and both the centrioles in the centrosome present perpendicular to each other. Understand? So now 13th question. Name the organelle which lies perpendicular to each other, in which has an organization like a cartwheel. So this centriole concept is very important for asking different competitive questions in different manners. So this is one of the different manner regarding the concept of centriole. Now let us see options. Option A, cilia. Option B, centriole. Option C, nucleus. Option D, flagella. So the correct answer for this question is option B, centriole. So both the centrioles in a centrosome arranged just perpendicular to each other like 90 degrees. It means L shape. 90 degrees arrangement can be seen to the centrioles in a centrosome. And looking like organizing with the example of a cartwheel. Okay, now moving to 14th question. How many peripheral fibrils doublets are present in a centrioles? Let us see options. Option A 9 plus 2, option B 9, option C 2, option D 0. So we need to identify how many peripheral fibril doublets are present in a centriole. So here the important point is nothing but 9. 9 evenly spaced peripheral fibrils of tubulin protein made structures are present in the centriole. Understand? So each of the peripheral fibril is a triplet, and the adjacent triplets are also linked. This is very important for competitive examination. And the central part of the proximal region of the centriole is also proteinous, and it is called as hub. Okay, now moving to 15th question, name the protein which is present in a peripheral fibrils. This area or this question asked in previous NEET examination, very important area. Option A tubulin, option B triosin, option C cysteine, option D serine. So the correct answer for this question is option A tubulin. As I told you, based on the centriole, different methods of questions, different ways of questions can be asked. This is one of the important method, but concept is same. So the peripheral fibrils of triplets made up of with a protein called as tubulin. Understand? So very important. Now moving to 16th question. Choose the central part of the proximal region of the centriole. Option A spoke, option B hub, option C nucleoplasm, option D core. So the central proximal region of the centriole, which is also proteinous, is called as hub. Correct answer is B. Next, 17th question. Pick out the organelle which forms spindle apparatus during cell division in animal cells. This question asked in last time K-CET examination, important. Now let's see options. A nucleus, B centrosome, C ribosome, D centriole. The correct answer is option D centriole. So let us see explanation. Let me explain in detail. The centrioles form the basal body of cilia or flagella. Remember it. And the spindle fibers that give rise shape to or gives rise the structures of spindle apparatus during the cell division in animal cells. It means even in our body also. So that is the main role by centriole during cell division time, producing spindle apparatus to divide the cell into two daughter cells. Understand? Now moving to 18th question. Identify which is not related to cytoskeleton involvement. A. Mechanical support, B. Motility, C. Maintenance of the cell shape, D. Cell division. So just now we had a discussion. Cytoskeleton is not involving in the process of cell division. Then who is involving for cell division process? Centriole. Then A, B, C are the functions of the cytoskeleton. It is not, D option is not the relevant to the function of the cytoskeleton. Next, 19th question. Choose the incorrect statement from the given. We need to identify incorrect statement from A, B, C, D. Option A. Both cilia and flagella emerge from basal bodies. B. 9 evenly spaced peripheral triplets are present in the centriole. C. Ribosomes are granular structures in the cytoplasm only. Option D is ribosomes units are S. S stands for sedimentation coefficient. The correct answer for this question is option C. It means they asked us to choose the incorrect statement. So the incorrect statement is option C. why it is wrong statement, how come it is, let's see. Ribosomes are granular structures, okay fine, but in the cytoplasm only. So ribosomes are not present only in the cytoplasm. Ribosomes are present even in the mitochondria, even in the chloroplast, even on the endoplasmic reticulum. So here C statement is incorrect statement. Next, 20th question, last question in this session. Name the cylindrical structures in centrosome. A. Ribosomes, B. Microbodies, C. Centrioles, and D. Nucleus. So the correct answer for this question is option C, centrioles. They are cylindrical in shape, called as centrioles, present in the centrosome. Both centrioles, it means two centrioles are present, both are perpendicular to each other. Understand? Hope you understood this video, this session. Thank you everybody.